Hello everyone. Welcome back to my channel. And today I'm going to do part 4 of my series on genetic treatments for neuromuscular diseases. And actually, this is going to be the first half of part 4 because as I started putting the slides together, I realized that this is kind of an involved topic, so I thought it was better to break it up into 2 parts. So this is about gene editing. You might have heard of CRISPR-Cas9, and so I'm going to talk about that and some general concepts on gene editing and sort of where we are now and what some of the limitations are. Okay, so to give a little bit of a review, uh, gene editing is distinct from gene transfer. Both can actually be considered as types of gene therapy, but in gene transfer, what you're doing is putting in uh, a new copy of a particular gene that's mutated. It doesn't do anything to fix the spelling mistakes that are associated with the mutation. It just puts another copy in that's correct and that will work. So the issue is that you're not making any corrections in what's already there. You're just putting in a new good copy. Okay, gene editing, by contrast, is actually fixing one of the spelling errors in the gene that's already there. And that's shown by this gold star. So, instead of 2 red X's and then you might add a gold star to it, this replaces one of the red X's, which is the gene with a spelling mistake that doesn't work, with a gold star, which is the corrected version of the gene which does work. Okay, now why are we only correcting one of the two? Well, it turns out that in most cases, people have, if they have mutations in both copies of a certain gene, the two mistakes won't be precisely the same one most of the time. And one of the things that is true of gene editing is that every particular edit can only fix one particular spelling mistake. So if you've got one spelling mistake in one copy of the gene and a different spelling mistake in the other copy of the gene, the two can't be corrected at the same time, which is kind of an important limitation. Okay, so uh, has been in the news a lot, and the proper term is CRISPR-Cas9. So, okay, CRISPR is actually an acronym, closely, regularly interspaced, short palindromic repeats. Um, okay, you don't have to remember that. That isn't terribly enlightening. But the reason that CRISPR, it turns out it exists in the natural world. And what it actually is the way that bacteria protect themselves from viruses. So bacteria can get infected by viruses just like we can. Now, what happens when we get a virus is we have an immune system that has specialized cells whose job it is to attack the virus or attack cells that have been infected by the virus. Okay, but a bacterium is just one cell. It doesn't obviously have an immune system, but it has, it turns out, a pretty clever way of fighting off viruses. So what it does is the virus reproduces by putting its genes into the genome of the cell that's infected, in this case a bacterium. But CRISPR is actually a way of breaking the virus's genes so that they no longer work. So if you say put an extra DNA base in, turns out that DNA bases go in groups of 3. If you put one in, then the grouping of 3 gets messed up and that inactivates the gene. So that actually works very well. And so the way that CRISPR-Cas9 works, okay, Cas9 is a protein that can cause a break in both strands of DNA. And when you have a break, most of the time it won't correctly repair itself to the way that it was before. So what CRISPR is a way of recognizing a particular DNA sequence in bacteria fighting off viruses. It's a DNA sequence that's typical of a viral DNA gene, so that when you see this certain pattern of DNA bases, that says, okay, that's not supposed to be there. I want to go there and create a double-stranded break, and most of the time it won't be able to repair itself correctly, which will inactivate the gene, so the virus is now neutralized. Okay, now it turns out CRISPR is not the first way of editing genes. Actually, before CRISPR came around, there were several others that I'd heard of in my work. A couple of the ones are what's called zinc fingers, and then talons, which itself is another acronym. But CRISPR actually works somewhat better, and because it has this guide sequence, genetic sequence, it's easy to program it to look for a particular DNA sequence to create a break in the DNA. Now, an important point is that in nature, CRISPR's function is to break genes, not to repair them. It's also true that it's always easier to break something than to repair it. So if we want to actually turn a non-functioning gene and make, just precisely the DNA change that will get it working again, rather than just some random DNA change that will break it, because it's already broken, there's no point, we're going to need to be a lot more carefully targeted than CRISPR is in nature. Okay, so a couple other points to finish up this, the first half of the gene editing section. And that is that every edit requires a different, or rather is targeted to a specific DNA sequence. And therefore, that's going to require a different version of CRISPR, or a different CRISPR drug, if you will, in order to make that particular change in DNA. And so that becomes a significant issue in a genetic disease where there isn't one very common mutation where a large, percentage of the people who have that disease have that particular spelling error in their DNA. The other thing is that the target sequence is going to basically make a DNA cut wherever it occurs in the genome. So you hope that the particular sequence of DNA bases that it's targeted only occurs once in a person's genome, or else you might have you might be clipping DNA somewhere that you don't want to. The other thing is, what if, say, all but one of the DNA bases are the correct ones? Maybe every once in a while it'll, bind to the wrong site and, make a DNA break somewhere that you don't want it. Okay, so there's a few things going on, and we need to basically adapt CRISPR as it naturally occurs to make a lot of the editing changes that we'd like to for genetic diseases. So in the second video, I'm going to talk about some instances where CRISPR, even in its naturally occurring form, can potentially be useful right now. Some of the other issues with broadly applying it, and then some of the adaptations that researchers are starting to make in CRISPR to make it more generally applicable. So anyway, thanks for watching, and I will talk to you later.